Hello and welcome to this eSeminar on the topic of sampling approaches for the analysis of foods, which includes the background to and examples from the approach adopted by the UK Food Standards Agency. This eSeminar has been produced as part of the Joint Knowledge Transfer Framework for Food Standards and Food Safety Analysis, and which is funded by the Department for the Environment, Food and Rural Affairs, the Government Chemist Programme for 2020 to 2023, the Department for Science, Innovation and Technology, formerly the Department for Business, Energy and Industrial Strategy, the Food Standards Agency, and Food Standards Scotland. The process of sampling can be thought of as taking a portion of a product, which is representative of the product in its entirety, and suitable for submission to a laboratory for analysis. It may be undertaken by enforcement authorities, regulators and businesses in order to provide assurance that food is both safe to consume and is what it is claimed to be. The testing carried out will depend on the purpose behind the sample in the first place and may include the determination of microbiological, chemical or other contaminants, as well as the composition of the test sample, nutritional parameters and authenticity. As highlighted previously, sampling may be undertaken by enforcement authorities such as Port Health and local authorities, regulators, which includes the Food Standards Agency and Food Standards Scotland, as well as by individual businesses within the food industry. There are multiple reasons why a sample could be taken, and these include to support enforcement action, to provide assurance on the safety or quality of a product for a business, as part of a research project to inform science and policy, e.g., through identifying the baseline level of a contaminant risk in a product, as part of a targeted sampling plan, acting on intelligence or hypotheses to detect potential emerging risks, or as part of annual regulatory monitoring in a specific area which is required by legislation, e.g. veterinary medicines. However, sampling can be expensive as well as resource-intensive and needs to be delivered in both a targeted and coordinated manner. Rather than analyze a product in its entirety, which is impractical, we use sampling to provide an indication of the most likely composition of a product with respect to the analyte being considered. This strategy has both advantages and disadvantages associated with it. In terms of advantages, it can provide assurance on the safety, quality, and authenticity of food and feed, and that it meets both regulatory and industry standards. It can also inform and support actions undertaken to address a specific risk of concern by providing both information on and evidence of a risk. In terms of disadvantages, it can only provide an indication of the composition of the product tested, and in reality, this may not be representative of the whole batch. Testing may only target a specific contaminant or microbiological areas, meaning there is potential to miss risks that aren't looked at. It can also be expensive, and it can take time to carry out the analysis, in which case a product may already be on the market, leading to costly recalls. A sample is a subset of the total population. It is important to note that when testing a product, you will only receive information on the sample tested, which may or may not be reflective of the wider population, the product on the shelves. It is for this reason that careful consideration is needed on why and what you are sampling, alongside the size and number of samples to be taken. The critical point of any sampling strategy is the number of samples that will need to be taken. As illustrated by the table shown on the slide, the more samples that are taken, the more likely an issue will be found. But, in reality, this needs to be balanced against cost and practicality. While there is no one-size-fits-all approach for sampling and the number of samples that should be collected, there are some general considerations that can be followed. These include the use of directed sampling, where there is a suspected or known risk, e.g. to support enforcement action, and conventionally utilizes small numbers of samples, depending on the relevant legislation, typically less than 5 samples. Surveillance sampling, which is typically employed to confirm a hypothesis or intelligence about a risk in a product and employs in the region of 30+ samples. Statistical sampling, which involves large-scale sampling in order to confirm to a high degree of statistical confidence the level of risks in a commodity, which will typically utilize more than 300 samples. When designing a sampling plan, there are a number of questions that need to be considered. These include the size of samples required, e.g. weight, volume, individual units. What retailers and businesses should be targeted? Is there regional variability between products? When is the sample to be taken? Do risks vary by season, weather? Is the food imported, as this may affect whether the product is available for sampling? Is the analyte being surveyed likely to be distributed heterogeneously? What action will likely be taken once the analysis has been completed? If the sample is to be used for enforcement purposes, additional considerations such as sample size and division will need to be addressed. Once a design for the sampling strategy to be adopted has been finalized, alongside the physical number of samples to be taken, careful consideration is needed on how the sample is taken and then stored before analysis in order to ensure accurate and useful results are obtainable. A good sample should be representative of the lot sampled, product batch, be taken and stored correctly to avoid cross-contamination, be stored in a way to prevent degradation, a reduction in quality of the sample, be appropriate for the analysis in question in terms of quantity taken and the purpose for sampling. Many of the conventional sampling strategies are designed to evaluate analytes that may be expected to be regularly distributed throughout a product. However, there are exceptions to this, an example of which is sampling for mycotoxins. These are a group of toxins which are produced by microfungi and that have the potential to cause disease and death in humans and other animals. Mycotoxins tend not to be distributed evenly across the product in which they occur, but tend to form local concentrations or hotspots. As a consequence, sampling for them in a product needs the adoption of a strict sampling plan to ensure that results are representative of the product. This typically involves taking multiple samples from across the product, which are then mixed by the lab to ensure homogeneity and then tested. However, targeted sampling is allowed where visible inspection of the product would identify a part of the product that may be prone to mycotoxin formation, for example, where the product has become damp and is showing mold growth. In addition to the points covered in the previous slides, when undertaking sampling and testing, it is useful to consider a range of factors on the analytical testing methods used. These include: does the sample allow for a sufficient limit of detection to be achieved and quantification to be able to detect the risk or hazard that you're looking for? E.g., when sampling soybeans for the presence of GMOs, has a sufficient number of beans been taken to achieve an LOD of 0.1%, which is equivalent to 1 bean in 1,000 beans? Does it provide a definitive conclusion on the presence or absence of something, or is it just indicative and may require further follow-up investigation and action? Is there any possible ambiguity with the results? E.g., if using ELISA, is there potential it may have cross-reacted? The figure on the slides summarises the current division of responsibility for food testing across the relevant government organisations. As the figure illustrates, FSA and FSS are responsible for general traceability, import control for food and feed, labelling, biological and chemical safety, and also biotechnology-related topics, which includes genetically manipulated organisms. The Department for Environment, Food and Rural Affairs, DEFRA, are responsible for import controls of animal-related products, labelling with respect to products containing animal tissue, composition and standards, but also for residues of veterinary products. The Department for Health and Social Care and the Health and Safety Executive are responsible for labelling of nutrition and nutritional health claims, pesticide residue, and composition and standards. There are three main regulations which set out how samples should be taken and tested. The Food Safety Act 1990, which stipulates the requirements for how samples should be taken and analysed for enforcement actions taken under the Act. The Sampling and Qualifications Regulations 2013, which outlines the qualification requirements for food examiners, public analysts and food analysts who interpret the results from enforcement samples. It also sets out guidance on certificates of analysis, which report the results of testing. And Assimilated EU Regulations 2017/625 set out the requirements for laboratories that test official samples. In addition, there is a wide range of legislation that sets out the requirements for the sampling and testing of specific contaminants and food products, such as mycotoxins and shellfish. These may require specific sampling approaches to be taken and or tests to be used by laboratories. So, why does the FSA undertake sampling? Sampling and subsequent analysis underpins the work of the FSA and related organisations, such as local authorities, LAs, in maintaining the safety and authenticity of the food supply chain. It therefore performs an essential function in providing intelligence and evidence on the safety and authenticity of food and feed on the UK market, assists in the support of any enforcement action to protect consumers, and meets the FSA's statutory obligations as a regulator. The FSA undertakes a number of forms of sampling for the analysis of foods. These main types are regulatory monitoring, where sampling activities are funded by the FSA and relate to a specific regulatory requirement, e.g. monitoring program for shellfish. Monitoring for science and research, where sampling is undertaken to inform a specific research and/or policy question, e.g. AMR. Surveillance monitoring, which comprises targeted unofficial sampling funded by the FSA to identify emerging risks and validate intelligence insights, with sampling priorities being identified via intelligence and expert insight. Official controls, where sampling funding is provided to local authorities, LAs, or port health authorities, PHAs, to undertake official sampling under the FSA's direction. This sampling targets specific commodities based on intelligence and surveillance sampling results. However, although this constitutes official enforcement sampling, there is no regulatory requirement for this sampling to be undertaken. Taken as a whole, the outputs from FSA surveillance sampling may be used to direct official control sampling, thereby making better use of limited resources. The FSA currently organises a range of surveillance and targeted sampling programs. These are Basket of Foods. This utilises the selection of a variety of commonly consumed food commodities analysed for a broad spectrum of compliance parameters. Sampling may be directed, for example, consist of most commonly consumed items in the average diet, or based on limited intelligence, or focused towards known characteristics of certain commodities that make them inherently higher risk. Detects emerging issues proactively and can be used for horizon scanning. Targeted retail surveillance sampling. This is a targeted program which, in response to intelligence or known emerging hazards, targets a specific area to establish if or the extent of any issues. This approach provides enough data for general monitoring, though not for rigorous statistical analysis, but can be used to inform further targeted sampling. Continuing from the previous slide on FSA sampling programs, there is also imported food sampling. This is a targeted program which tests hypotheses in response to intelligence or known emerging hazards. Sampling may be more reactive than pre-planned to accommodate any new intelligence. Again, this approach provides enough data for general monitoring, though not for rigorous statistical analysis. Statistical sampling. This involves a larger scale targeted sampling and analysis. Targets are driven by the current needs of the organization and known gaps in our understanding of the risks associated with specific foods. The sample size and breadth of data collected is suitable for meaningful statistical analysis on which policy and regulatory decisions can be based. The FSA provide extensive guidance and best practice on the undertaking of sampling for the analysis of foods. The examples listed on the slide are not exhaustive, but provide a selection of the outputs from recent sampling campaigns. This eSeminar has been produced as part of the Joint Knowledge Transfer Framework for Food Standards and Food Safety Analysis, and which is funded by the Department for Environment, Food and Rural Affairs, the Government Chemist Programme for 2020 to 2023, the Department for Business, Energy and Industrial Strategy, the Food Standards Agency, and Food Standards Scotland.